is X-Talk's webinar. Today's talk is entitled SDTM in Real Time, a Case Study. Initial datasets by first patient in, final datasets within 24 hours of study lock. My name is Andrew Jernan, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, so please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen, and if you require any assistance, please contact me at any time by sending a message using the chat panel. Now at this time, all participants are in a listen-only mode, and the presentation slides will advance automatically for you. And please note that this event will be recorded and made available for future download. At this point, I would like to thank Omnicom, who has helped develop the content for this presentation. Omnicom Systems is dedicated to helping the world's pharmaceutical, biotechnology, CROs, research and medical device organizations maximize the value of their clinical research investments through the use of innovative and progressive technologies. Omnicom's electronic data capture and eClinical technologies have been used in almost 4,000 clinical trials around the globe. Another thanks goes to Alchemies. Alchemies is a leader in innovative medicines that address the unmet needs and challenges of people living with debilitating diseases. As a fully integrated global biopharmaceutical company, Alchemies applies their scientific expertise, proprietary technologies, and global resources to develop products that are designed to make a meaningful difference in the way patients manage their disease. Now, I would like to introduce our first speaker for today's event, and that is Keith Howells. Keith has 20 years experience in the application of information technology to the drug development process. As Senior Vice President of Development at Omnicom, he's responsible for innovation for the TrialMaster product line. Keith's prior positions were Head of Development for Metadata Solutions and is Head of Development for Oracle Clinical. Our second speaker for today is Prashanti Vanuva. Prashanti has 6 years experience in the pharmaceutical industry. As Senior SAS Programmer at Alchemies, she's mainly responsible for creating SDISC, SDTM, ATOM, and DEFINE documents for FDA electronic submissions, and also providing oversight into data management activities like data cleaning, building CRF, and edit checks. She previously worked as contract senior SAS programmer for large pharmaceutical companies and in all therapeutic areas, and has also worked as a research associate at Wayne State University in the NINDS-funded laboratory. And now, without further ado, I'd like to hand the mic over to Keith Howells. Keith, you may begin when ready.
Great. Thank you, Andrew. Hello, everybody. Again, my name is Keith Howells. I hope we have pretty interesting topics for discussion today, all around SDTM. This will be my piece of the presentation. First, we'll give a lightning overview of what this standard means, and then we'll drill into a couple of domains or data sets that are specified in this standard, and some of the mapping challenges you have trying to get the source data into the required format. There's also a need to make sure that any coded fields are managed by controlled terminology, so we'll talk a little bit about how that works. And then what we see as the technical approaches to producing SDTM, and we'll pause for a little survey, see what all of you online are doing. And then we'll move specifically into the TrialMaster EDC product and look at the mapping capabilities there. So first, an overview of SDTM. It's a standard for data submission maintained by CDISC. It defines standard domains. A domain is basically a data set, and each of those standard domains has standard variables. And the names would be very familiar to you. There's a demographics domain, ComMeds domain, AE domain, Vitals Labs, and so forth. Now, if you have a data value that isn't one of the standard variables, then those go into what are called supplemental qualifiers, which is broadly a normalized variable name, variable value kind of structure. So it's basically an extensible standard where the things that are expected to be collected have a home, and then the other variable goes into one of these supplemental qualifiers. Now, today the FDA is recommending that you provide SDTM data sets along with submissions. Most recent year, about a third of submissions have SDTM data sets, and that helps speed their review process because they have some really fine tools to reanalyze the data, look at patient subsets, look at efficacy and safety variables, and so forth. Now, under PDUFA 5, the Physician User Fee Act, that will become mandatory. The process is that there is supposed to be a draft set of standards. It's actually currently about 6 months late, so the industry is eagerly awaiting that. There'll then be a comment period, and once the comment period is closed, there will be a 2-year compliance period. So best we can estimate it's about 3 years out before this will be mandatory, i.e. any submission in 2016 and beyond will require SDTM data sets for all the studies in the submission. So if a study going on today is going to be submitted in the future, you will be required to have SDTM data sets with them. In terms of the physical delivery, they're delivered as SAS transport files. There's also an explanation file called define.xml, which basically defines the structure, and then an annotated CRF so that the reviewer can map each variable back to where it was collected in the protocol. Just some metrics. The latest version is 3.1.3. The implementation guide for that is over 300 pages. So it's a large body of work, needs a lot of explanation of how to populate all these variables. There are 34 domains averaging 23 variables each, so that's about 700 pieces of data to be populated. And if I could summarize, not easy. Any of you who have actually had to produce some of these in production, I'm sure could associate with that. It's not something where it's a routine programming task and you could send it out to a team that wasn't medically knowledgeable. It will tend to come back incomplete and with errors. So let's take a look at a couple of examples. Let's start with demographics because that's a fairly simple one. On the left-hand side, we have a representation of how this might be collected in EDC, and on the right-hand side, the structure of pieces of the demographics domain. So you can see we have a study ID, what domain it is, demographics, a unique subject ID, and then birth date in a prescribed format, age and age unit, sex, M or F. So here's where we start to see the code list coming into play, ditto for race, ditto for ethnicity. But broadly here will typically be a one-to-one mapping with how you collected it, and therefore not terribly hard. Now let's look at a questionnaire. This is the top part of an SF36 questionnaire. And you can see you're asking a number of questions in various categories. So we have a sort of general health question, a general rating, how it was compared to last year, and then limitations of activities and other sections underneath not shown. So this is typically how you'd lay it out in EDC because you want to only show the appropriate answers to each of the questions via, in this case, these radio buttons. But in SDTM, what they want is basically to normalize this as question text, question value. So this would come out where the first one, in general, how is your health, as very good, how is your health compared to 1 year ago, about the same, and so forth. So what you need to do here is a transpose, turn it from the way it's structured in EDC into this test name, test value normalized format. Third example is vital signs. This is more of a hybrid where it's like this. We got test code, test result, and test units, but we have, for instance, this position parameter, which is only relevant for some of the variables. Your blood pressure varies according to whether you're sitting, standing, or supine, but your body temperature doesn't. And therefore, the position is only expected for those particular variables. So a medium complexity rotation program here. And then also not shown here is calculation into standard units. So if you collected temperature in Fahrenheit and standard units of Celsius, then you would need to have that calculated standard measure for temperature in the data set. Let's move on to talk about control terminology. These are actually managed by the National Cancer Institute and used by CDISC on an OEM basis. Some of these code lists are fixed, so you have to use only the entries in there and no more. But many of them are extensible, which means you have to use the ones that are there, but if you have additional values, you can put those in as well. Our strong recommendation is to collect the data using CDISC code lists in the first place, or at least code lists with a one-to-one mapping to CDISC. Let's take an example, the action taken on an adverse event, which is marked as a not extensible code list. And you can see on the right, we've got various things that you can, actions you could take. You can increase the dose, you can not change it, reduce it, you can interrupt or withdraw it. And these are, at the time, the same thing. You stop giving the patient the study medication, but obviously drug interrupted. At some future point, you resume dosing. Let's say in your protocol you haven't captured that, you're not interested in that, and you've just got drug interrupted or stopped. You've now got a challenge to map that to one of these CDISC entries, because you're basically going to have to go look at the start date of the adverse event, look at all your dosing forms, and figure out if you could see a dosing record after the adverse event start date, in which case you're going to mark this as interrupted rather than withdrawn. So this is one example where if you just sort of outsource and say, hey, figure it out, go write me some code, you're likely to get answers that are wrong or not what you expect if you have got this kind of fuzzy definition of your data in the first place. So now let's look at the technical approaches for producing these. The top is the classic extract, transform, and load, where you run your study in EDC, you produce raw data sets, and then you just write procedural programs. Most common tools are SAS or SQL that collects the data and does various formulae calculations and then produces these data sets. The alternative we're floating here is to actually do it all within the EDC tool and map your data to where it belongs in SDTM right within the EDC database, and then as part of just general outputs from your study, producing those SDTM domains. So in terms of comparing approaches, the first one, the pros, well, it's technology neutral. It doesn't matter what EDC tool you're using or any other tool, any other sources of data, as long as you can produce those raw data sets, you can follow this. It's obviously proven, because for many years it was the only way of doing it. And it can be outsourced again with the caveats that this is a higher skilled process than your regular programming job. In terms of the cons, you have to redefine the metadata. The first thing in writing these programs is to figure out what your code lists are and your variables are and their lengths and all these types of things, because you don't know anymore. You've just got a set of source data sets. That makes it both time-consuming and error-prone. And because you've got procedural code which you're sort of editing each time, that's not reusable across studies. You're going to have to validate those programs each time. In terms of option 2, reuses the metadata because you're in the same database. So, what are my variables? What are the code lists against them? How are they grouped? How long are they? All of that's available. Our experience is that this is faster and you can test early, because as soon as you've got data in these data sets and start running that against your analysis programs, it's validated with the database and it's reusable across studies, because if you copy the study, then all the metadata comes with it, including your mappings. In terms of cons, it does therefore require all the data in EDC. So if you're not sending, say, your lab data into EDC, you can't produce the lab domain. PK data similar. So you may be going to be loading data into EDC just to be able to get it out again. As far as we know, TrialMaster is the only product that supports this, so it is specific to one particular EDC technology. And as you'll see, there is still some programming required. So given there are these fascinating two ways of doing things, we're going to pause here for a couple of quick survey questions to find out what each of you are doing.
Well, thanks, Keith. And you're certainly correct here. Here's the first of two poll questions we have for you, and you can vote on these in real time. The first question is, what approach is your organization using to produce SDTM data sets? And your four options that you can vote on right now are: we are not currently producing SDTM data sets, we write programs while the study is ongoing, or we write programs after the study is locked, or we have a tool for producing SDTM data sets. That question again that you can vote on in real time, I encourage you to do so right now. Just click on your screen there. What approach is your organization using to produce SDTM data sets? And with that, it looks like most of you have voted. I'm going to close polling and share all the results with you. So there we are. We can see that the majority of you said we write programs while the study is ongoing. That's followed at 29%, saying we are not currently producing SDTM data sets. 11% said we write programs after the study is locked, and just 4% of you said that you have a tool for producing SDTM data sets. So I'm going to close that and actually launch the next poll for you, and you can again vote on this in real time. This is the second and last poll. This question: how quickly are your SDTM data sets available after study lock? And I encourage you to vote on that right now. Click on one of those options. Your options are equal to or less than 1 week. Your second option, equal to or less than 1 month. Third option, 2 to 3 months. And your fourth option is greater than 3 months. And again, that question is, how quickly are your SDTM data sets available after study lock? And it looks like most of you voted, so I'm going to close that poll again and share the results with you. And there are the results. Looks like the majority of you at 45% said equal to or less than 1 week. Again, followed at 27% by equal to or less than 1 month. 9% said 2 to 3 months, and 18% said greater than 3 months. And with that, I'm going to hand it back over to you, Keith.
Great. Thanks very much, Andrew. Some quite impressive results there. Those nearly half doing it within a week of study lock. So congratulations. Let's move on to the mapping overview. So we're now starting to get into some of the mechanics of doing this within, again, the TrialMaster EDC tool. Over on the left-hand side, we've got your classic EDC structures: what you know about the trial, what you know about the site, your patient number, and whatever. And this reference start date, which is the start date of dosing, which we calculate back onto the patient record, obviously from trawling all of the dates in the exposure forms. Then say we've got our adverse events, we've got our demographics and so forth. And on the right-hand side, I'm just showing some of the variables just for space reasons, just showing them for the adverse events domain. So how do we populate those variables? Well, the study ID will typically come off the trial form, so maybe one-to-one mapping with the trial number. The unique subject ID is typically some kind of concatenation of the site number and the patient number. Sometimes it includes the trial number as well. For most of the variables, those are just going to be collected variables. So you can see AE term goes to AE term, LLT to AE LLT, and so forth. In terms of the tool, where the name matches, it will map automatically. Where the name doesn't match, you will have to do that by hand, and we'll see that in a tick. And then we've got this one at the bottom, this AE study day, which is going to be a calculation between the reference start date and the AE start date. And so, again, you're going to be doing little fragments of programming, but it's just a SQL expression here that's just going to be a date difference calculation between those two dates. So I've got a couple of screenshots of physically what it looks like. So this is Trial Builder. This is Study Design Tool. This is a Windows tool, so you get multiple windows, drag and drop, and all those good productivity things. You create something called an export template, and then you say, I want to map it against the standard, and you get shown the standards that are supported. The definition standards is just metadata, so we have other databases where we've implemented 3.1.3, the cancer domains, the new device domains, and so forth. The beauty is those just get loaded in the database as the target of the mapping, and then you again map that to your collected variables. So having clicked that, it will then say, okay, here are all the domains I've created for you, their short names and so forth. And then you would click on one of these ellipses to drill down, and that's when the hard work starts, and now you're mapping these individual variables. So here is just some of them. You see our scroll bar is halfway down. Here are the variables. We color code them to let you know whether you have to give an answer for these or not. So you would work on the orange and the yellow ones. The white ones you're allowed to have empty or not mapped. And you can see some of these have auto mapped. You can see the serious code and the action taken, the relatedness are all populated. But this one at the bottom, the start date, is not populated. It's mapped to empty string. That would fail validation if we left it that way. Well, why is that? Well, on the left-hand side, we can go look at our study metadata, and we can see, well, we just called it ASTDT. So we were missing this C on the end, so that's why it didn't auto-map. So what do we do about that? Well, we basically just click on it, and we drag it to where it belongs, and then it would be mapped. So basically, as simple as we can make it, here's what I want, here's where it goes, and you're done. If you wanted to put in a SQL expression, again, for this study day calculation, you would click on these ellipses, and you'd be dropped into a window where you could basically write that SQL expression and validate it. So hopefully that gives you a feel of what it looks like. In terms of the overall capabilities, on the current production version that the Alchemies are using, it will do the auto map based on the item name, the drag and drop mapping as I just showed, SQL expressions for computed variables, and sequence number calculation. There's a lot of these domains where you have to number these things and then reference those numbers in other domains, so we can do all of that. We're working on another version that will come out towards the end of this year that will do some of these more complicated operations like the transpositions I showed for the questionnaire domain, union, so if you've got multiple questionnaires, you can union those sets of data together. Aggregate functions, so min, max, average, so again to calculate things like the reference start date, and to be able to calculate things into one domain and then use those as source objects for other domains. So those of you used to procedural programming, that would be the equivalent of creating some kind of temporary table or staging data set that you can then pass into another set of calculations. So today, for those domains that need capabilities that the current tool doesn't support, we implement those on the back end using SQL. So today, there's some percentage of the domains we implement using SQL, and all the rest are implemented using this mapping tool. This next version coming out towards the end of the year, we expect to be able to do 100% of this using the mapping tool. So to summarize, SDTM is recommended today. It will be mandatory within about 3 years. You should design your study items and code lists to be compatible with the standard, or you're going to have far more work than is appropriate when you try to produce these data sets. There's two technical approaches, the extract, transform, or load, or the map within the EDC tool. And we believe this mapping is pretty easy to use because it's got intelligent defaulting and this very elegant drag and drop interface. And with that, I'll hand over to Prashanthi to describe how that's worked out in practice.
Thank you, Keith. Okay. Hello, good afternoon, everyone, and good morning to American US people as well. Today, this is Prashanthi Vunnava, and I'm going to present how we do SDTM mappings within TrialMaster using TrialMaster export functionality, and what are the advantages of having this SDTM within database, and what are the advantages we really had by having this during the beginning of the trial, and what kind of activities we have done within database lock. So Alchemies has been producing CDIS standardized SDTM datasets from using this TrialMaster export functionality that's been developed by Omnicom. for all our regulatory submissions from past 2 years. This whole webinar will cover all the SDTM mapping specification, how they are created during the study design, and also the benefits of having SDTM build during the conduct of the study and having final data sets within 24 hours of database lock. Basically, we follow CDAS standards to collect the data, and based on CDISC SDTM related standards, we create the SDTM data sets, which most of our studies are being done that way, and which helped in submission of clinical trial data to FDA without any issues. So what is CDISC? It's Clinical Data Interchange Standards Consortium, and it has been, it's a non-profitable organization which has been established to support the data collection, exchange, submission, and archive of clinical research data and metadata. Good standards are more essential for any success of the clinical trial because they help in ensuring the data collect is complete and accurate, and it also helps in for submission to FDA without any problems. And at the same time, it helps in designing the study protocol, data collection, analysis of the results, reporting through regulatory submissions, and also archiving clinical data electronically. What is CDASH? It's Clinical Data Acquisition Standards Harmonization. It is also being a collaborative approach of CDISC to develop the content standards for data collection in all the CRFs. This standard is based on SDTM. So that's why whenever you do data collection, it is most important, if you are doing any kind of submissions, have an SDTM data sets that are CDISC compliant. At the same time, follow the CDASH standards in collecting the CRFs. What is SDTM? It is a study data tabulation model which is designed for standard regulatory submission report of case report tabulations from all clinical trial studies. So prior to SDTM export utility, what we have faced earlier before coming to Omnicon and using the trial master utility, we had so many problems with basically a lot of companies does not follow CDAS standards in collecting the data on the CRF, and at the same time not following CDISC standards to create the SDTM data sets. The main drawbacks of doing programming at the end of the trial, which is labor intensive, which is also time consuming, and it's error prone, and it's also non-repeatable, and it lacks quality, and also at the same time it loses traceability. Traceability is more like a CSR to analysis data sets to SDTM to CRF. FDA highly recommends having a direct implementation method, having from direct approach from CRF to CSR. Cleaning of the database is also complicated when you don't have standardized SDTM data sets. When you do submissions, it's going to have more compliance checks that will report as an error when you submit in CEDAR in FDA submissions. For example, end of study day should be later than all AE and conmet start and stop dates. End of study day means disposition status of CRF. So traceability, I'll discuss more in detail about traceability. How this is highly recommended by FDA, and it helps in the logical flow of the software development life cycle. And it complete traceability from CSR back to ADAM to SDTM to CRF must be developed. That's the most direct approach to have that way of designing a CRF to the creation of CSR. Coming back to Trial Master SDTM export functionality, what are the advantages in having this export functionality when compared to other vendors? So Omnicom has developed an extensive library of CDAS standard electronic CRFs in collecting the data, which helps easily for formatting the CDIS SDTM datasets within the trial master database. Hence, we started using Omnicom Trial Master Export Facility for all our study mappings within database during the beginning of the trial. And we tried to work with other vendors, but it was really cumbersome to in creation of SDTM datasets using mapping tools, but there were several drawbacks that are less flexible, and it's very costly, and it's also time consuming. The quality is very low, and also another disadvantage is not having coding within the trial master database. Right now, Omnicom Trial Master Export Facility have its own auto coding functionality, which you can do coding within the database. That will make your later on the processing easier to create the SDTM datasets, because in SDTM datasets you have coding for adverse events, converts, and medical history. So it's good to have a coding. It's more advantageous than having outside of the database. Most of the companies, they do outside of the database, which is cumbersome to get back that data into trial master import functionality, and it's going to again create a problem for building the SDTM datasets. What are the strategies in creating the SDTM datasets right now using the trial master import facility is following all these points. First, what we do is generate SDTM mapping specification in Excel file. That will be done by Omnicom SDTM expert, who does all the mapping specifications for us, and she creates an Excel file and she sends it to us for review. What does the mapping specs contain? Each protocol specified assessment or RF must be mapped to a proper SDTM domain. Some of the standard domains that include special purpose domain, which are demographic, subject elements, and subject results. And the interventions domain, example, exposure, converts. And events domain, which is adverse event, disposition, and medical history. And another domain which is findings domain, which includes labs, vital signs, ECG, inclusion exclusion criteria, and questionnaires. And the last one is supplementary domain. This supplementary domain is additional data, whatever that cannot be mapped in the parent domain. Like for example, in AE you have a question saying, did subject discontinue due to AE? So you don't have a parent variable that's available in the parent domain. So that kind of information you can create in a supplementary domain. But this, for some of them it is fine, but not. You should be very careful in designing the CRF itself. When information is not needed for, it's better to avoid that kind of information, not to capture it and have it to map it to a supplementary domain. FDA, even in this implementation guideline, it clearly says try to avoid as much as you can to create a supplementary domain. Have information, all information available in the parent domain. Only if it's necessary, create a supplementary domain. And the final one is the trial design domain. Trial design domain, which will be more based on the protocol. You need to summarize, like trial summarization, like what kind of, which phase of this trial is, and what is your primary objective, what is the age limit of the study protocol, all that information. And the trial visit domain, which will have all the visits for each subject. And trial arms, which will include all the treatment information. Trial inclusion exclusion criteria, this is more like a protocol designed inclusion exclusion criteria mapped in the trial inclusion exclusion domain. And then make sure all the imports coming into trial master database are compliant with CDISC control terminology. Example, labs and ECG. Because usually labs and ECGs are not done within the site. They are being imported into the trial master because as they are done outside of the database. All the sampling, like even PK, they're all done outside of the database. So when we do that kind of information outside of the database, we need to make sure they're compliant with CDISC control terminology, like some test codes, like lab test codes and the ECG test codes and the ECG test and lab test. Also, try to avoid as much as having a control terminology with that's not compliant with CDISC. Like having in the, for example, in the CONMED form, you have a CM dose field where the units are captured there. Usually the site will have its own free text field. Try to avoid those free text fields so that they cannot enter, and provide them a control terminology list where they can pick from that list instead of having a free text. And the fourth point is reviewing and approval of the mapping specs by the programming team. Initially, everything, the mapping specs was being created by Omnicom SDTM expert, but the review and the approval of the mapping specs is more like whoever the client, sponsor is responsible for reviewing them and make sure they are mapped correctly. And then SDTM datasets will be released into UAT for review by the team, which is UAT is more like a user accepted testing. As everyone know, for data managers, they do UATs and going into live and all that. Make sure all the CRF variables are derived variables are being mapped in the SDTM. Derived variables means in terms of in demographic domain, you have reference start date and end date. You make sure they are derived correctly. And then when you are when you checked everything, all the mappings are being done correctly, then go for a run an OpenCDISC software, which is available freely to download, and you can anyone can download that, and you can run your checkings on your SDTMs, and you make sure no errors are found. For sure, all the errors should be handled when you run a report. If you find any errors, try to correct them within the database. Also, make sure the warnings are being reviewed, because some warnings you cannot ignore. So, like for example, control terminology warnings. If there is no code list in the control terminology, as you know, everyone knows that the SDTM implementation guidelines, they clearly explain there is extensible list of code lists. So they don't have everything in the code list. So in those cases, you can assign your own client or sponsor defined control terminology list. Any issues identified during the UAT will be fixed, like errors definitely have to be fixed, and warnings you need to, some of them you definitely need to try to fix them. And then when they are fixed, and again they are released into another cycle of UAT. And after those fixes are done, you can release the database into production, which can be every time whenever, whenever you go into the EDC database, you can just pull down the SDTM dataset from there using an export functionality. Also, anytime during the trial, if you notice when the study is ongoing, if you notice any issues in the SDTM, then it can be fixed by assigning SDTM expert on it and tell them what the problem was, and it can be fixed using a mid-study change and make necessary modifications and then again release into production. So most of the complicated ones we deal in the SDTM, they are right now being fixed using SQL scripts, as Keith already explained how they use SQL functionality within the EDC trial master database. So that's how we do the mapping strategy in metadata-driven process. So I would like to explain clearly how you map the control terminology code list when you have imports coming outside of the database. For example, in this case, lab import specifications, we create specification document ahead of the study trial. And when the protocol is ready, you basically create a specification document so that the vendor test names and codes that are being imported into the trial master will be mapped to the SDTM variables. So we have at the back end of the system our own SDTM code list, like this SDTM code and SDTM name. Those are being mapped to lab test code and lab test name, which are according to the control terminology. And these are the test code and test names that are being used by the lab. So, so you need to make sure your specifications are correct and they are being imported the way you want it. So you need to make sure that even the, for example, even PK samples also done the same way. So I explained earlier that there are derived variables in SDTM that have to be done within the database using SQL scripts. So for example, in this case, this is the best example to explain that. Example, drug accountability log and exposure domains are created for the purpose of capturing daily doses and in-clinic visits. We need to take into consideration both the forms to derive the start date and end date of dosing. Example, reference start date. Reference start date is RF SDTC, which is the minimum date and time available in both drug accountability and exposure forms will be start of the dosing date and time. In terms of reference end date, maximum date and time available in both drug accountability log and the exposure forms will be end of dosing date and time. And if lab dosing date is captured in the CRF, then it will be mapped in the demographic domain to reference end date. So I clearly explained with an example for reference start date how it is mapped. For example, in this case, you have a drug accountability form and an exposure form. So all the visits, visit 2 day 1, visit 3 day 8, visit 4 day 15. These are the dispense date of the drug and the amount and the return date. So the start of the dosing will start on day 1, which is the first date. But in exposure form, you have in-clinic visits. Basically, when the subject comes in on day 1, he gets the dose. On that day, he will take the dose in clinic. So that is being captured in the exposure form, where with the time being captured. So when you have time and date available, that's your reference start date. Even though here you have the same visit being dispensed on same date, but you don't have time. So you map the reference start date based on that, based on the exposure form. So final conclusion, Omnicom export functionality helped us build very high standard SDTM datasets, which helped us immediately after the start of the study. We were able to build them and then review them and release into UAT and release into production, which helped us a lot in our company. Targeted metadata can streamline process and can be reused across studies. So it's not like just like having data available, but the same mapping can be used in different trials as well. Like example, safety, they can be easily mapped across the studies. With SDTM domains made available earlier in the study, we were able to do data cleaning using these data sets throughout the trial, and if any problems arise during the data collection, they will be identified and caught earlier than at the end of the trial. And also, SDTM data sets available so early, Alchemies was able to log the study within 24 hours after database lock. And this is a tremendous, very important step in clinical trial, in the design of the study. The reason is because when you have as soon as the lock is done, you have data sets available. That makes easier for making the decisions on that trial. Like for example, as Alchemies is a public limited company, it was very useful for us to have the top line result available for higher management to make the decision whether making the decision on the results, and also in creating the analysis data sets and tables, listings and figure, and delivering all the results within a week. And I tremendously had very advantages in using Trial Master and having data sets available within 24 hours for study log. That's it. Thank you.
Well, thank you very much, Prashanti, and I'd like to thank you both actually for those presentations. Very insightful. And at this time, I would like to invite our audience to continue sending their questions and comments in right now using the questions window for the Q&A portion of this webinar. I've already received some of those questions, so I'm going to start with those. Our first question here: does the production of SDTM datasets have any impact on system performance?
Let me take that. The short answer is no. Behind the scenes, the EDC data is being replicated to a separate server, and all of the processing, the reformatting, and the production of the data sets happens on that separate server. So we make sure that the server that is supporting the interactive use of EDC is not involved in this more high-powered processing work to produce the SDTM data sets.
Right, okay. Can SDTM mappings be copied between studies?
Again, short answer is yes. Because it's just the study metadata rather than procedural programs, it's all in a part of the study design database. So if you extract some or all of that for use in another study, those mappings will travel with it, and so you wouldn't have to redo that mapping work for the second study.
Okay, thank you. Our next question is for Keith again. And this attendee says, the FDA has requested that the actual SDTM field length be restricted to the max length of the actual data. Is this implemented in the Omnicom SDTM export tool?
Oh, good question. Yes, that's the #1 complaint from the FDA is the physical size of these things. And one of the downsides of SAS transport format is it's actually a fixed character format. So if you say this is character 200, but there's only 2 characters in it, it will actually store 200 characters worth of space. And these things just get very slow to move around and open and so forth. So when obviously we dimension these for the maximum allowed length, which is indeed 200, but as you're mapping, you set that length according to the variable or the calculation that's populating it. So it's part of the process to set the lengths right for your character variables at the same time as you're doing the mapping.
Okay, thank you. This next question I'm hoping maybe both of you could comment on. And that is, does one need to use C- or SDTM standards for annotating of CRF in paper trials and EDC trials?
Prashanti, why didn't you take that first, and then I'll comment your answer.
I would highly recommend to have SDTM annotations in paper trials as well as EDC trials, because that makes easier for a reviewer in FDA to have SDTM annotations on an annotated CRF.
Okay, Keith, any further thoughts there?
Yes, so basically if you start with C- standards, the automatic work is done for you, or more of it is done for you. But no, you could have any variable names you like, any item names, any code lists, and just the automatic mapping will not give you any help at all. But you can then, through the drag and drop or SQL expressions, map your study into the target. So Yeah, the bottom line is using C- will help the process, but it's not required.
Great, thank you. This next question is for Prashanti. An attendee was commenting 1 or 2 slides back from the end of your presentation. Were you talking about the C-Disk validator? Is it meant there?
Yes, that's correct. This OpenCDisk is a freely available software to download, and you can just download and you can put all your whatever the data that's already being created. You can import there and run that OpenCDisk software program, and then it will show up all your errors and warnings. And it will cover most of them. Maybe a few of them won't be covered, but still it is a good validated software to check your SDTM data sets, whether they are good to go for FDA submission or not. And Alchemies uses that software all the time.
Right, okay, thank you. Another question here. Could you comment on the production of define.xml and annotated CRF?
Sure. So those are 2 other deliverables. The define.xml basically defines the structure of the data sets and where the variables come from, whether they're derived or collected and so forth. And then the annotated casebook is basically a printout of the source CRFs, and for each of the places where the site user can enter data is an annotation of where that lands in the SDTM datasets. We do both of those on a service basis, so they don't quite come out automated from the tool. Things like the annotated CRF, they tend to get kind of messy with lots of little where clauses around. And so we haven't got that fully automated, but we do that on a service basis.
Right, thank you. Another question here. Any difficulties in getting third-party vendors, lab or ECG, etc., to submit data in C- or like standard for mapping, or would one need to import an interim map?
Prashanti, do you have any insights on say, your lab vendors and how easy it is to get the data in a format that you can load?
Yeah, right now we did not have that many problems. When you have your specifications mentioned correctly in the beginning of the trial itself, you will not see any errors during the ongoing of the trial. I never experienced any kind of problems. Usually, morely it's reconciliation between EDC, what's entered in the database, to what's being imported into the database by lab vendors. Currently, we did not have that many problems. Even though there were problems, it was minor fixes, and it can be fixed very easily. It depends on the specs also. If your specs are clear, and there should not be any problem in importing lab data into EDC.
Great. Sorry, please finish.
Yeah. Even PK is the same way. PK should not have any problem in importing into trial master.
So just from the mechanics point of view, there are 2 primary ways that third-party data can be loaded in. One is a file import, where you can just ask the vendor to provide a delimited ASCII file. So there you don't really have to know what the element names are, what they're calling them. Just say, look, I want the lab test code in column 5 and the value in column 6 and the units in column 7, please. And then we define that as what we call an import template, and then files matching that template can either be manually loaded in the system, or we have a listener where the vendor can just leave those files on a listening directory, an FTP directory, and they get loaded in automatically. The other mechanism is real-time through the application program interfaces, say for patient diary data that I know Alchemies have used. comes in real time through application program interface calls. There, you do tend to need to harmonize the element names because that is now an ODM message where the vendor is providing form names, group names, and item names, and they have to match the equivalent names within TrialMaster.
Right, thank you. Our next question. So when SDTM was already available, what was the reason for C- to be introduced as data collection guidelines from CDISC?
Well, C- I can answer that question. Well, C- is more like it's a CDISC led innovative tool. That's why if you use C-, basically it's more like you are having a CDISC compliant SDTM datasets. So when you are collecting the CRF itself have C- standards, which is CDISC standards that will apply to SDTM as well.
Great, thank you. Another question here. Can the cross-domain derivations be defined?
Short answer is yes. You can either do that in the mapping tool. You can drag and drop these things from a variety of sources, and the mapping tool will figure out the database joins it needs to do under the covers to implement that. Or for some, it's more pragmatic to do the derivation in the EDC tool. So I mentioned the reference start date. That can basically be trawled within the EDC tool, find the earliest one, and just plop it on the patient form or the demographics form so it can be used from there. So a choice of tools. Again, the mapping tool is drag and drop with SQL expressions, or if you do it through the EDC tool, then you're programming that in JavaScript.
Okay, thank you. What about deletion of some variables you may have in there? Can you comment on that?
So basically, you don't. If there's a standard variable in the domain, you don't delete it. You just don't map it, assuming it's not a required or expected variable. We do have the ability to add your own variables and/or your own domains. So I guess if you add one and then decide you don't need it after all, you can delete it. But if it's in the standard, then you have to have it there. You just don't map it.
Yeah, I can comment another note on that one. If you have that variable, I would suggest to not delete it, then map it to a supplementary domain if it's really needed. If it's really not needed, then you can delete it, but it can be captured in the audit trail of TrialMaster database, which if FDA comes back why you deleted that variable, you can have a note to file why you deleted that variable, whether it's needed, if it's explaining why it's not needed.
Right. Okay. Our next question, how do you create the define.xml? Do you have an XML programmer, or is there a program to create the define XML?
A bit of both. Our goal is to produce that 100%, but the spec keeps getting enhanced, so we're just a little behind on that one. So there is a first cut that comes out of the mapping tool, and then that just gets supplemented by yes, someone doing XML programming. Yes,
I would also add a note on that. Yes, definitely a tool is needed, and you can use the OpenCDISC validator tool to create a define.xml, and an XML programmer has to program it, edit it in order to have the format FDA wanted.
Okay, thank you both for that. Our next question, I think we're just about out of time, so this will probably be the last question. If the length of the variable can only be 40 characters, are we allowed to define the content of this variable in SAS up to 200 characters?
Yes, you are, but again, that's the kind of thing that would irritate the reviewer, because you are guaranteed to have at least 160 characters of empty space in the final data sets. That is, again, making those data sets bigger than they need to be.
Right, okay. Well, thanks for that clarification, and thank you both for those answers. We've reached the end of the question and answer period today. And if you do have further questions, please direct them to the email addresses showing on your screen. For Keith, that's khowells@omnicom.com, and for Prashanti, that's prashanti.vunnava@alchemies.com. Thank you, everyone, for participating in today's conference. You'll be receiving a follow-up email from XTalks with recorded access to the archive for this event. And a survey window will be popping up on your screen, and your participation is appreciated as it'll help us improve our webinars. Now, please join us in thanking our speakers today, Keith Howells and Prashanti Vanuva. We hope that you found this conference informative. Have a great day, everyone.
Thank you.